Let's look at the element zinc. Zinc is normally found as a +2 cation that may be associated with other ions. Zinc is the 24th most abundant element in the Earth's crust. Zinc concentration in uncontaminated soils ranges from about 70 to 160 micrograms zinc per gram of soil. It is found in the soil solution. Zinc content in soil created from basic igneous rock is normally high. Zinc is an essential catalytic component of over 300 enzymes. This is where you get your metabolic function. Zinc is involved in many enzymes. Zinc is extremely important in the enzymes such as carbonic anhydrase, alcohol dehydrogenase, zinc copper superoxide dismutase, aldolase, and fructose 1,6 bisphosphatase. If you look at those, fructose 1,6 bisphosphatase is the basis of glycolysis. Therefore, zinc is an essential catalytic component for all living organisms on planet Earth. The uncatalyzed conversion of carbon dioxide and bicarbonate is slow and does not meet metabolic demand. Carbonic anhydrase catalyzes the reaction where zinc hydroxide picks up carbon dioxide to make zinc bicarbonate. Water comes in and releases bicarbonate to the system. Then this cycles over again. If this is allowed to occur naturally, it is slow. The enzyme is located in the cytosol and in the chloroplast. Bicarbonate is very important because it is used in many reactions according to the metabolic requirements of the organism. Processes which may require bicarbonate or carbon dioxide are found in C4 plants. PEP carboxylase directly uses bicarbonate. Alcohol dehydrogenase is an enzyme where zinc is bound by 3 ligands to the protein. At one site, a water molecule is absorbed. This will allow for the conversion of aldehyde to ethanol. This is a very important process in metabolism. It is common in roots. The copper-zinc superoxide dismutase is a complex of about 30 kilodaltons required to detoxify the superoxide radical. Superoxide is a very potential chemical that is going to react with a lot of different things. Therefore, it should not be found in the plant. This is a detoxification type enzyme. It is located in the stroma of the chloroplast. Superoxides are produced by the uptake of electrons by oxygen. 2 superoxide radicals combine with the 2 hydrogen ions, producing hydrogen peroxide. The hydrogen peroxide needs to be broken down because it is a very reactive chemical. Sunscald, which is a destruction of photosynthetic membranes by oxygen radicals, leads to severe loss of fruit and vegetable quality. Sunscald occurs in plants where there is an accumulation of peroxide. Resistant species such as tomatoes, peppers, and cucumbers have high concentrations of superoxide dismutase. This particular enzyme complex requires zinc and is extremely important. These are just a few examples of what zinc actually does. Method of uptake. Uptake is driven by the plasma lemma hydrogen pump and requires ATP. Copper inhibits zinc uptake. Cation uptake, which includes calcium, cadmium, and zinc, across the tonoplast into the vacuole is a hydrogen ion antiport. Therefore, it is driven by the pH difference between the vacuole and the cytosol. This is the way you get intake into various parts of the cell. Zinc is not bound to stable ligands as the case of copper, nickel, and iron. VA mycorrhizae can substantially increase the uptake of zinc. Plant concentrations in the range of 20 to 100 micrograms zinc per gram of dry matter are sufficient for the plant. It doesn't take a lot of zinc in the soil in order to be able to have sufficiency. Seed zinc content remains constant even with high concentrations in the soil. Basically, seeds already have a certain amount of zinc in them. That is what they need in order to be able to germinate. They do not pick up zinc out of the environment. If you get a zinc deficiency, it's going to be in soils with high pH. High pH is going to be over 6.5. Below 10 to 15 micrograms per kilogram zinc in dry matter is an indication of a zinc deficiency. High levels of phosphorus may induce a zinc deficiency. All of these ions seem to have a pecking order of which gets absorbed first or which inhibits what. That's why you have to balance your different elements. Phosphates may affect the physiological availability of zinc in the plant tissue. Phosphates are very important in zinc metabolism. Phosphorus-induced zinc-deficient maize plants were found to accumulate high levels of iron. That's a compensatory mechanism. Zinc-deficient soil is cultivated in the cropland of about half of Turkey and India, a third of China, and most of Western Australia. This accounts for 30% of the world cropland. Zinc deficiency does not occur in certain areas. These are zinc-deficient corn plants. You can see the chlorosis that is occurring. When you look at this, you may go, wait a second, is this chlorosis due to nitrogen? It looks very much like a nitrogen deficiency. Or could it be an iron deficiency? Or is it a zinc deficiency? The only way you can tell is to take tissue samples and have them analyzed to find out what the zinc concentration is. This is on avocado. You can see how this is appearing on the younger leaves. It looks to be very problematic. You're getting a distortion of the younger leaves. They are not growing up to the morphology that you would want. Luxury consumption. If there is too much zinc in the soil, it will be absorbed and deposited in the older leaves. This will immobilize it for a little bit of time, but eventually these older leaves are going to drop off and recycle the zinc back into the environment. Toxicity. Toxicity of zinc is interesting. This is peanut. The stem has a reddish brown or a purplish discoloration to it. You can see the leaves are not quite as green as they should be, which is a decreased chlorophyll level. Remember, the decreased chlorophyll level could be due to a nitrogen deficiency or an iron deficiency. The reddish-brown discoloration sometimes shows up with a phosphorus deficiency. In here, we talked about phosphorus and iron all being related to zinc. This becomes important if you have a toxicity like this. What you need to do is look at concentration in plant tissue in order to determine what really caused that. Where do we get zinc from? It comes from the breakdown of parent material in the soil. Zinc is added to the soil primarily through the weathering of rock. Zinc uptake in crops is usually less than 0.5 kilogram per hectare per season. This is quite small. Zinc application may depress the uptake of cadmium. They looked at this back in the 1960s to see what might remove some of the material that could be toxic in the soil. Zinc is an important element. Doesn't take much of it. It is required everywhere by plants and by higher organisms as well.